This podcast is brought to you by the School of Advanced Study, University of London. All of our podcasts are available from our website, www.sas.ac.uk.
So, it's a great pleasure to be here again. I'm full of praise for Barry's impromptu performance, which should have impressed you far more about the nervous system than anything I'm going to say. The title of my lecture means, of course, I'm going to talk about vision, which has been my major interest in my career, and it is the sense that we generally consider to be dominant in our experience of the world. We believe that our eyes and our brains working together provide us with a full and reliable, veridical description of what's happening around us. We trust it, we depend on it, we guide our actions on the basis of it. The other senses, of course, are important, but for most of us who have vision, we feel that vision is, as it were, the central sense on which the others are hung. I hope in this lecture to show you that vision depends much more on what's already in our brains than on the information that's coming in through our eyes, that it is frequently unreliable, that the impression that we have of living in a constantly present, always present world, that we're just passing in a window of time through a world that's always there, is an illusion. I'll show a number of examples that I hope will convince you that you have reasons to doubt your visual experiences, even though that's not what those experiences tell you intuitively. So I want to start, I hope, with a movie that's going to work. I'd like you to watch this.
I'm Richard, this is Sarah, and we're going to perform the amazing colour-changing card trick with this blue back.
Sorry, that's for recording, it's not projecting.
Now the idea is very simple. I'm just going to spread the cards in front of Sarah and ask her to push
It's a card trick. If you watch the card trick, I'll explain what he's saying. She's choosing one card. They're all blue-backed cards, and she's been asked to choose one of them.
Okay. Now,
Just watch
Sarah
carefully.
could have selected any card at all from the deck.
Okay, she's taken one card.
Which is now face down on the table. And what I'm going to ask her to do is show us which card she selected.
Okay, so she's showing the card to the camera. Oh,
The three of diamonds, okay, excellent choice.
Three of diamonds.
That card goes
Back into the pack.
Now I'm just going to spread the cards face up on the table.
Face
A
up.
little click of the fingers. And you'll see that Sarah's card here has now got a blue back.
It's blue on the back, as all of them were.
Slightly more surprising is all of the other cards have got
They're all red.
red backs.
Well, it's a nice little
Amazing colour-changing
card trick. I don't know whether any of you noticed anything strange about the movie. Some of you might have seen this movie before. While you were watching it, 4 other changes of colour took place. Did anybody notice them? I've seen it before. So we're going to replay the video from a different viewpoint, so you can see what was actually done. It's exactly the same video, but now from a different camera, viewing the same
We're
scene.
going to perform the amazing colour-changing card trick with this blue-backed deck of cards. Now the idea is very simple. I'm just going to spread the cards in front of Sarah and ask her to push any card towards the camera.
Okay, let's see. I'm going to go for this card here.
Okay. Now Sarah could have selected any card at all from the deck, but she selected the card which is now face down on the table. What I'm going to
Remember,
ask
you've seen this movie before, just shot from a different angle.
Right, so the card that I chose was in fact the three of diamonds.
The three of diamonds, okay, excellent choice. That card goes back into the deck. Now I'm just going to spread the cards face up on the table, do a little click of the fingers, and you'll see that Sarah's card here has now got a blue back. Not particularly surprising. What's slightly more surprising is all of the other cards have got red backs. And that is the amazing colour-changing card trick.
So this is somewhat alarming. You viewed the whole scene, you watched it forensically, you were looking for the trick in the card, the trick in the changes of the cards. And while that happened, other events, dramatic events happened, which most of you, perhaps virtually all of you, were entirely unaware of. Now that should be a bit disturbing because we rely on our vision to guide our actions. We need to know that what is there now was similar to what was there in the past. Say you're called into a court of law to give testimony about something that happened. What would you say if you were asked to describe, well, what colour was the background in that scene that you saw? You've just had demonstrated very convincingly that you can't remember what the previous colour was, or at least you can't tell that it's different from what you're seeing now. Let me show you another example, hoping against hope that it works, and it's not going to. This is absolutely extraordinary. I've replaced every movie while you were watching me, listening to Barry's efforts, and all of them appear to have reverted to images. What I can do, I think, is just the best that I can do is this. I just hope that I'm going to play it directly, exactly. All right. If you watch the dot in the middle of this display, you're going to see these coloured dots constantly changing. Every dot's changing constantly. Just keep viewing the dot in the middle. What happens when the array moves? Keep watching. So when they're static, when the display's static, all the dots are constantly changing colour. And while they're moving, are they not changing? Okay. Watch them, repeat it again, but watch an individual dot out in the periphery here. Don't look at the dot in the middle. Try and track the array of dots as they move. And to the extent that you can hold your fixation on them, they are still moving. The array is exactly the same all the time. And I'll show you another example. Again, look in the middle. All the blobs are changing size. And when they move, it apparently stops. So here's another example on a different time scale of things that are changing, in this case while the scene is moving, not being detected as having changed. At the end of the lecture, I want to try and explain to you what's happening and then examine the significance of that for the apparent reliability of visual experience. So now if I can go back to the presentation, this one perhaps now would have worked. No, that's again static. It's remarkable. All right, how have people thought about vision in the past and how it worked? Go back to this year, 1664, which turned out to be a rather significant year in the development of ideas about the brain. It was the year in which Thomas Willis published that book that I mentioned in my last lecture, the Cerebri Anatomy. And it was also the year of publication of another extremely influential book, even though the author of the book had died some years before. It was the French translation of the Tractus de Homine by Descartes, who died in 1650, died 14 years before. The French edition was distributed much more widely than the Latin edition, was read much more widely because French was the language of the educated classes across the whole of Europe at the time. Yes, it's going to be one of those days, isn't it? I ought really to turn off the Wi-Fi, but all right, too complicated. This book was remarkable in that it postulated an extraordinarily mechanistic view of the way in which the human body worked. I'll just show one quotation. All the operations that I've attributed to this machine, and he was talking about the brain, waking, sleeping, reception of lights and all the senses, the impression of ideas on the common sensorium. This was a classical view that the senses were combined initially, and then thoughts and rationality were derived from that combined view of the senses. The retention of the ideas in memory, appetites, passions, movement of the external members of the body. I wish you to consider all of these to follow naturally in this machine from the disposition of its organs, although neither more nor less than the movements of a clock. So he's comparing the brain, including the human brain, with the functioning of a clock, that one action followed another automatically. Incoming sensory information provided, as it were, the energy for the process, the initial stimulus, which then triggered actions and reactions, passing, as he thought, through the fluid in the brain, and then out through the nerves to activate muscles. Descartes had viewed the retinal image himself, and he reports that work in La Dioptrique. He wasn't the first person to see the retinal image and to understand how the eye functioned, but it must have been pretty impressive to a philosopher interested in the theory of knowledge and epistemology to view a dead eye of an ox that he'd got from the local butcher around the corner, in which he'd opened a window in the back of the eye, cutting away the sclera of the eye, putting a piece of paper over the jelly inside the eye, holding it up to the window in his room, and seeing a little tiny upside-down image, perfect little image of the window projected onto this screen in this dead piece of matter from an animal. To someone interested in how we understand the world, that must have been a riveting experience. And he imagines himself looking at the image, and the optics are drawn accurately with the lens of the eye forming an inverted image focused on the retina. He, in his mechanistic view of how the brain might work to generate actions and appetites, movements of the limbs, memories, and so on, he suggested that the pattern that had been set up on the retina, the image that had been set up on the retina, was somehow propagated isomorphically, retaining the same characteristic, the same form, into the brain. And this is one of the illustrations from the French edition, from the Traité de l'Homme, showing his view of the two eyes, each with their own image of the external world, each having an inverted image. And there's a problem because there are two eyes, but we see only one world, and there is only one world. He imagined them passing in perfectly coherently, like through the fibers of a coherent fiber optic bundle, and then projecting onto the surface of this thing, which is one of the ventricles inside the brain. Because at this stage it was thought that the fluids in the brain were where the processing of information took place. So what he imagined was that the eye delivered, as it were, a picture onto the surface of the ventricles, and that the vibrations, or whatever mechanism the nerves used to transmit their impression from the retinal image, were then passed through the fluid of the brain, causing actions in other nerves that moved eye muscles or moved the hands, the arms, whatever. In addition, and you see in this illustration particularly, he considered the problem of how this robotic machine, which was doing its job simply on the basis, as he said, of the disposition of its organs, of its parts, how it could also have impressions and awareness of the world. He did not, and some people have accused him of it, he certainly didn't think that simply the presence of, as it were, a picture inside the brain solved the problem of how we see the world. he says very clearly in La Dioptrique, we shouldn't imagine that it is by virtue of this image in the brain that we see the external world, because to do so, we would need another set of eyes inside the brain. There would be a sort of infinite regress, if you like, of eyes looking at eyes inside the brain. Just having a picture is not enough, any more than, you know, having a photograph of your loved one in your wallet, in your pocket, enables you to see that person. If it's hidden from your view, then you won't understand it. So there has to be a different mechanism for our knowing about the outside world and having what looks to us like a picture of it. There must be a different mechanism other than having an apparent picture inside the brain. And the solution to that, as well as the resolution of the difficulties they saw from a religious perspective in imagining that human beings were simply machines, the solution to all of those problems was, of course, dualism, the notion that in parallel with the mechanical actions of our bodies and brains, there was a second level of activity involving the mind or the soul, which was privy to, as it were, a second view of all the information that was passing through the brain and directly experienced it. And that soul was us, the possessor of subjectivity, the owner, as it were, of the brain and the mind that the brain generates. So here's his view of where that interaction took place in a structure called the pineal gland. He was impressed with the pineal gland. He dissected. He wasn't as skilled a dissector as Willis, but he certainly dissected brains and saw that most of the structures of the brain come in twos. It's a bilaterally symmetrical organ. So, you know, there are two cerebral hemispheres, and there are two of most things, two halves of the cerebellum. But there is a single distinctive structure of which there's only one in the middle of the brain called the pineal gland. It's a gland that secretes a hormone called melatonin. It's involved in regulating the diurnal rhythm of activity in the brain, the sleep-waking pattern. But he didn't know that. But he thought that the fact that it's unpaired, it's singular, and its placement quite close to one of the major fluid-filled ventricles of the brain puts it in a strategic position to eavesdrop on things that were flowing through the fluid, and therefore simply to know about them, for the soul to be informed about them. So here, the ripples that are passing through the fluid don't just go off and do their thing and activate nerves, they also impinge on the pineal, very conveniently converging in such a way that only a single pattern of activations corresponding to the external world is created, despite the fact that it starts off with two sets of ripples. They converge on each other, so the image becomes fused into one again, and equally conveniently re-inverted, so that the soul sees the world the right way up, rather than the inverted retinal image. This notion that there's a picture in the brain, but of course it can't be the picture that does the job, has run through sensory neuroscience, and it's still, it's still superficially attractive. The idea that a spatial relationship between bits of activity in the nervous system might somehow represent spatial relationships in the external world, and frankly, it does. By virtue of the connectivity between different parts of the brain, activity in one place is likely to activate one particular set of actions, activity in another place, a different one. So the arrangement with which activity arrives in a particular bit of brain can become reflected in the reactions to it, just as Descartes said, almost mechanical reactions to an incoming pattern of information. I mean, an extreme example, the right side of your visual field, because of the arrangement of the anatomy, projects to the left side of your cerebral hemispheres. And the left side of your cerebral hemispheres is mainly responsible for controlling the right side of your body. So your right hand is activated by your left hemisphere, and your left hemisphere sees the right side of visual space. So the two things are coordinated. Things that you see over there, by relatively local connections within a single hemisphere, will cause actions of your hand to grasp and so on. It doesn't, as Descartes recognized, explain the subjective experience of seeing the world, but it is a very convenient anatomical arrangement to represent spatial relationships. Far from the eye transmitting information by vibrations, which was Descartes' favoured view, and also that, by the way, of Newton 50 years later. The eye works principally electrically. If you look into the eye with an ophthalmoscope and you ask the person whose eye you're looking into to look at the bright light of the ophthalmoscope, as an optician might do when they're examining your eye, then this is what you, the viewer with the ophthalmoscope, will see. You're looking down into the eye, onto the surface of the retina of the eye at the back. It's got blood vessels running over it, supplying oxygen and glucose to the superficial layers of the retina. The back of the retina is supplied by a different blood supply. You can see a darker patch in the middle, and that corresponds to the part of the eye called the fovea of the eye, which we point towards things by making our eye movements. We point that little darker patch towards things of interest as we look around the room. That's happening all the time. It's darker because it has a pigmentation in it, the macular pigment, and you'll notice that the blood vessels avoid that central area, at least in the normal state of the eye. The blood vessels are scattered around the rest, but they avoid the very centre, and that's part of a whole range of clever tricks to ensure that the image falling on the very centre of the eye is of the highest quality. This white arc you can see at the side here is just part of a circle, a bright disc called the optic disc, and that's in fact a perforation in the back of the eye, a hole in the back of the eye, which is filled with 1.5 million nerve fibres leaving the eye. The crucial cells whose fibres leave the eye are called ganglion cells, and they're scattered over the whole of the front surface of the eye, about 1.5 million of them in humans. And all their fibres stream across the front of the eye and then plunge down through that hole, bundle together and form the optic nerve, which goes off to the brain. So keeping numbers in mind, about 1.5 million channels of information are passing back to the brain. And they have to provide all of the information from the whole of the eye. Now, if we look at the anatomical arrangement in that middle region, the part that you point towards things, it has distinctive features which, as I said, seem to be directed at producing as good an image as possible and sampling that image in as detailed a form as possible. This is a cross-section through the central region of a human eye. The front of the eye is down here, so the lens is there, it's forming an image, the light's coming through and forming an image. Immediately you see there are lots of different little circles here. These all represent nerve cell bodies. There are lots of different types of nerve cells. The retina of the eye is not a simple structure, it's quite complicated, and that reflects the fact that embryologically it's an outgrowth of the brain. The eye is really in its origins part of the central nervous system, it's part of the brain. You see a number of layers, and perhaps the most distinctive layer is the one at the back here, the very back of the retina. These curious cells with long processes that stick up, those are the photoreceptors. The long things are processes that contain pigment that catches light. So that's where the crucial process takes place, the transduction of light into an electrical response. Now, out here in the edge, away a bit from the central fovea, the pit, fovea means pit, there are distinctive layers of cells. And you can see that they're pushed aside in the middle, and this is thought to be an adaptation to avoid optical aberrations that might be produced by the light having to pass through these other layers of cells before it strikes the receptors. The receptors are at the back, which seems a bit strange, really. You'd think for the best possible interpretation of the image, you'd have them at the front where the image is being formed, but they're at the back for all kinds of reasons, mainly embryological reasons. In addition, they have to be in contact with a pigment at the back here to regenerate the pigment each time it's changed by light. So it has to be that way around. Okay, so within this central region, you can see another adaptation which is distinctive, and that is that these receptor cells are thinner and longer, more closely packed together. There are about 70,000 receptors in that very central region, which, by the way, represents a patch about 2 degrees across, 2 degrees in angle. Visual field, I mean, the whole field is 360 degrees. That central region that you point towards things is 2 degrees. And how big is a degree? Well, it's about the size of your thumbnail at arm's length. So So if you imagine this central patch, which is so crucial to your vision, the thing you point towards anything of interest in order to scrutinize it, is equivalent to about 2 thumbnails across. That's all. It's a little thing that's bobbing around in the visual field that you're actually looking at things with. There's other stuff happening in the peripheral retina, but the density of receptors is lower, the optical quality is less, and there are a lot of other differences which we'll come to in a moment. So this is already sounding a little bit different from the way we see the world. What we're seeing inside the eye is an enormous amount of anatomical heterogeneity. This highly specialized region in the middle, very different arrangement at the edges, different characteristics, different resolution of detail, different capacity to detect color and contrast, for instance. And yet, when we look at the world, it doesn't look like that. You don't get the feeling that whatever you're looking at is sharp and clear and the rest fades off into the periphery. It seems superficially to all to be the same. And so it would be deeply disturbing if you thought that the room you're in was actually fading off in color or in detail. I mean, our visual system is not there to tell us about the defects in its own optics. It's to provide the best possible description of what's actually happening in the outside world. But it makes you realize that a lot of your impressions of what's happening around you are literally not possible because the limitations of your eyes limit the information they can provide to the brain in ways that your visual experience don't inform you of. Okay, so the arrangement of the eye suggests that the central region, small region, is extremely important in sampling information, and it's moving constantly. The photoreceptors themselves are a remarkable piece of adaptive biology. First of all, they're incredibly small. Those narrow processes in the very center of the human fovea are about 1.5 micrometers across, 1.5 thousandths of a millimeter across, which is as small as cellular processes get. So they're amongst the smallest cell processes anywhere in the body. And they're absolutely packed with this very sensitive photopigment that catches light. And the light is guided by the shape of the photoreceptor. It is guided through the photoreceptor to optimize the chance that each photon of light will be caught as it goes through this stack of pigment before coming out at the end. Not all of it is caught. Some of the light that goes into the eye, that reaches the retina, passes through the receptors and bounces out, isn't caught on the way out, and comes back out the front of the eye, a small amount of the light, but most of it's caught. Some crucial experiments in the 60s showed just how sensitive the photoreceptors can be. This was work done on the toad, originally on the toad retina, but it's now been done in mammalian eyes. We know, including the human eye, from biopsies taken during surgery on the eye. What you're seeing here is in isolation, in a suspension in tissue culture, a piece of the retina of a toad, and you can see one of these photoreceptors sticking out here. That's the back of the eye. This thing is a glass tube connected up to a recording amplifier. And suction can be applied to the tube to suck the process up into it to get maximum contact with it for electrical recording. And here it is with the photoreceptor sucked into the tube. Since it's glass, light can be shone onto it. And in one of the crucial experiments done by this particular group, the minimum amount of light needed to detect a response was measured by filtering the light down to the point at which, on average, it contained only 1 photon, the minimum quantity of light energy that can be emitted. Now, any of those amongst you who know anything about physics know that it would be impossible to deliver reliably 1 photon at a time. You can filter a light until, on average, it might have 1 photon, but because of the quantum indeterminacy, there will sometimes be 2 photons in the light, sometimes none at all, occasionally 3, very rarely 4, and so on. They'll be distributed in a Poisson distribution. So what they did was to expose the photoreceptor, record electrically from it while exposing it to lights that, on average, contained just 1 photon of light. And this is one of their original recordings. The blips at the bottom correspond to the time when the light flash was turned on, when the shutter was opened. Remember, the light flash contained, on average, 1 photon. And these are the electrical responses that 1 photoreceptor produced. Well, if it produced a response, sometimes it did nothing at all. Sometimes it produced a just detectable small thing here, sometimes a thing about twice as big, and occasionally something perhaps 3 times as big. And if those amplitudes were plotted out, then, and the frequency of occurrence of the 0s, the 1s, the 2s, and the 3s was determined, it followed perfectly a Poisson distribution. So it's almost certainly the case that the smallest of these events corresponds to the electrical response generated by one photon being absorbed, this one 2, this one 3. The photoreceptor is giving a detectable response when it catches the smallest possible unit of light energy. It is as sensitive as it possibly can be. That's really remarkable. And we know from completely different experiments in human beings that under ideal conditions, when the wavelength is just right, when the eye is fully dark adapted, human beings can reliably detect a dim light against a dark background when it contains as few as 7 to 10 photons. 7 to 10. The whole human observer can detect a light when the flash contains only 7 to 10 photons. Well, you might say, well, if your photoreceptors can detect 1, why can't you detect 1? And the answer is almost certainly that if the brain were to set its threshold that low, then it would make mistakes constantly, because the photopigment, the chemical molecules that catch the light, are unstable. They have to be unstable in order to respond to light, I summarize, to change their configuration when they catch light. But because of that instability, every now and then, one of them breaks down in a way that mimics exactly what it would do if a light photon had been caught. So by setting a threshold of a few events rather than just one, the brain is less likely to make mistakes and to see non-existent lights. So the photoreceptors are as sensitive as they can be, and remember that they're scattered out across the whole of the retina. They're more concentrated in the middle, but they're everywhere. So in a sense, the photoreceptors, the 8 million or so cone photoreceptors, the 120 million rod photoreceptors, which you use at night, those are the really sensitive ones, they are viewing the whole of the image that happens to be on your retina, and each of them is responding if there is sufficient light to cause such a response. And indeed, if there's more light than the threshold amount, they will give a graded electrical response. So they're providing information about the amount of brightness at each point on the retina. As I mentioned last time, they're pixelating the retina almost in the way that the elements of a digital camera do. So in that sense, it sounds a bit Cartesian that there is an image on the retina. It's being transmitted as a kind of image to the brain along all these photoreceptor channels, then condensed into the 1.5 million ganglion cells. You're going from 130 million photoreceptors to 1.5 million ganglion cells, an enormous compression of number into the optic nerve and off to the brain. This is what it looks like, simplified. Here's a cross-section through the eye. Here's the fovea in the middle here, and the nerve fibers go out of the back, and here's the junction where they leave the eye. Horace Barlow, who was my PhD supervisor, very eminent neurophysiologist, was the first to record electrically activity from these nerve fibers. Remember that the nerve fibers which are going out are already some distance from the receptors, so they've gone through several stages of other nerve cells and a lot of processing. So when he recorded from those cells in the frog retina, he would have expected, if there was a simple relay of information, that each of those nerve cells would respond in the same way that the receptors do to light, and that their responses would be graded in some way with the amount of light. What he actually found was something much more complicated and much more interesting than that. What he found was that if the retina was illuminated with very small spots of light, then indeed the ganglion cells, these are the ones with their axons, did respond to the local brightening of their group of photoreceptors. But if the spot was expanded to include other photoreceptors, the ganglion cells stopped responding. So it was responding only to local illumination, not to illumination including neighbouring receptors. So some clever processing was going on, which he called lateral inhibition. And the way that he interpreted it was as a device for compressing information. Essentially what the eye is doing is cutting out or attenuating information about areas of uniform illumination. If you look, for instance, at a bright blue sky with a flat horizon, let's say you're in the desert and you're looking at a flat horizon, dark sand, blue sky above, then it appears to be uniformly blue and bright and uniformly dark and orange. In fact, your retina will be providing very little information from within the body of the sky or the body of the sand. Your ganglion cells, your retinal cells around the junction between the sand and the sky will be very active, but not the others. So it's spatially differentiating, as a mathematician would say, spatially differentiating the image, transmitting information only about the edges. Those of you who are old enough to remember the introduction of Xerox machines, the first photocopying machines, you will know they had an annoying habit when you tried to photocopy a photograph of turning it into a kind of line drawing. And in a way, that's what the retina does. It's transmitting information only about boundaries and edges because of this phenomenon of lateral inhibition. I mentioned in my last lecture that process of restricting the information and describing key features of the image is suddenly heightened when the signals reach the cortex of the brain, the cerebral cortex of the brain. So the work by David Hubel and Torsten Wiesel, recording not from the retina, but from the cells in the visual areas of the cortex, the first visual area of the cortex, showed that they respond. These are you'll remember from if those of you who were here for the first lecture, this is a burst of nerve impulses. When one of these nerve cells is shown in front of the animal's eyes, a line of a particular angle. But the distinctive feature at the level of the cortex, which does not occur earlier, at least not in higher mammals, is that the cells are selectively sensitive to the angles of lines. So here this cell responds very strongly to an oblique line, but not to a vertical line or a horizontal line. Different cells respond to different angles. So you see the beginning of a process whereby the brain appears to be, yes, operating broadly as a kind of pictorial representation, spatially distributed information, but within that array, what it's doing is looking what these individual cells are doing, are looking for key features, for features of the image, which then activate the cells and passed on for analysis, presumably elsewhere in the brain. I mentioned last week that one of the most important discoveries in the last 30, 40 years has been the fact that that vision depends not just on a single area, the back of the brain, as was thought by 19th century neurologists, but on a whole array of areas. And during evolution, more and more of these areas have been added. So perhaps 40 or 50 of them, distinct areas in humans, with some indication that they're performing different sorts of computations. I think I showed this example in my last talk, using results from functional magnetic resonance imaging to provide an indication of where activity is increased for a particular kind of visual stimulus. So if you show people static images while they're in the scanner, you get an increase in blood flow, because that's what fMRI detects, at the back of the brain in the classical visual areas. But if you show them moving patterns like this, and you take the difference between activity produced by that and that, you subtract that from this, then you see additional activity in this area Semyonov called V5. The Americans call the middle temporal area, an area much further forward here, which we know from work in monkeys particularly is devoted to the analysis of visual motion, movement. Well, that led to the view that, and Zeki's name is associated with this idea, that there is division of responsibility, division of labor, if you like, for analysing the visual scene between all these different areas, different modules, each of which contains tens or hundreds of thousands of nerve cells, and each module has a certain architecture of local connections which allow it to compute different aspects of the visual scene. Motion in this area, but other features in other areas. Well, an obvious question to ask, particularly with reference to the motion area, is how efficient the computational process is. If these areas are there to inform your awareness of characteristics of a scene, then they've got to accomplish various, essentially computational tasks. A very obvious one for the motion area is this one. How does the brain tell the difference between movements of the retinal image which are genuinely produced by movements of objects in space and movements which result from movements of the eyes? Because the movement on the retina can be indistinguishable in those two conditions. A student of mine, Loredana Santoro, a few years ago, looked at this question using that particular visual display generated on a television screen shown to people while they were lying in a scanner. And their task was always to look at the cross in the middle of the array. The whole of the rest of the scene was darkened. All they could see was these bright lines. And I think this is going to be one of my movies that doesn't work, even though it says that it should. But I can, in fact, it says that it is. Isn't that annoying? In the first condition, so-called passive motion, the bars moved from side to side while the cross stayed still in the middle of the screen. So the retina was being stimulated by movement, the bars appear to move, the person was looking steadily at the middle of the screen. And this is the classic arrangement for producing activity in MT, and here it is. This is the resulting differential activity produced by that stimulus, massive activation in the whole of this area, so-called MT plus, MT and the surrounding area. But she then changed the arrangement, and instead of moving the bars from side to side smoothly, she moved the cross with exactly the same time course. So the bars were static on the screen, and the cross was moving, and the person was instructed to track it. So if their tracking was accurate, it reproduced exactly on their retina the same movement that was present in the first condition. Bars were moving just the same way, but in this case, the movement of the retinal image was being produced by them moving their eyes, not by the bars moving on the screen. And remarkably, that does not activate MT. Marginal, insignificant activation. So somehow MT is able to do that little bit of algebra that's necessary to subtract out, if you like, the effects of one's own eye movements and derive a signal, or in this case a lack of signal, which tells you not what's happening on your retina, but what's happening outside. So the first indication that the brain is basically trying to compute what's happening in space rather than what's simply happening on your sense organs. And the final condition that she looked at, which is even more critical, was what she called tracked motion, in which the cross moved and the bars moved together on the screen. And the person tracked the cross, the bars moved with the cross. Therefore, if the tracking was accurate, then the bars didn't move at all on the retina. But they appeared to move because she was following them, just as my hand appears to me to move. Even though I'm tracking it, it's always in the same place on my retina. So this is really a critical test of the hypothesis. And here's the answer. MT was activated, or at least a subdivision of MT was strongly activated by that stimulus. Stimulus in which there is virtually no retinal motion, but there is active engagement of the eyes, and in which motion of the external world is seen. So it really looks as though MT is providing, at this crude level anyway, a signal which is related to the perception of the world rather than just the retinal stimulation. And there are many other good examples of that. This is an illustration from one of Semiozeki's early popularized presentations of his results when he was describing areas responsible for different function. And in this case, this is actually, I think, a result from a PET study, positron emission tomography, in the early days before fMRI, showing different regions of the brain. Here's the hemisphere, the boundaries of the hemisphere here, and two spots of activation, that's MT, generated by looking at moving black and white patterns. But a different region was activated a little bit further back in the brain, closer to the primary areas, when the stimulus was one of colour compared with black and white, coloured patterns compared with achromatic patterns. And he concluded that this area, which he called V4, was responsible for responding to colour, whereas MT was responsible for analysing motion and didn't seem to be interested in colour. Well, what this implies then is that different components of what you see to be singular and bound together and coherent are actually being analysed in very different parts of your brain. This poses a problem that cognitive scientists recognised a long time ago, and philosophers too, the problem of binding, how the brain puts together the impressions that result from analysis that's happening in very different places in the brain. How, for instance, when you recognise the form of an object, how do you know that the colour of that object belongs to that form? There must be a process by which the colour is, as it were, added to the neural representation of the form, not to mention the motion. And we know that that process of binding of colour can go wrong. There are the very elegant experiments done by Anne Treisman 20 years ago or more now, showing that if people are exposed to a series of coloured shapes, a blue square, a red triangle or whatever, and then are asked immediately afterwards to recall what combinations of colour and form they've seen, they sometimes mix up the colours and the forms. The colours appear to belong in memory, to belong to one of the other forms that they saw. So colour seems labile in that sense. It's not reliable in its attachment. I think that the fact that colour, well, for two reasons, colour is special. Colour is special from an evolutionary perspective, because there's very good evidence that mammals other than monkeys and human beings do not use colour habitually to determine their behaviour. I won't say they're unaware of colour because, for instance, a cat can be taught to make colour discriminations, but it takes thousands, literally tens of thousands of trials giving a food reward for responding to the right colour to train a cat to make fairly reliable judgments of colour, whereas a monkey on a good day will learn to do it in half a dozen trials. And that reflects the fact that cats have only two different visual pigments in their cones, a blue catching one and a green catching one. That's the minimum that you need to make colour discriminations. But what's happened crucially in primates is that they have evolved a third visual pigment that catches at the red end of the spectrum. They have three pigments. That extends their total sensitivity of light more broadly across the wavelengths to include red, and it gives them an enhanced capacity to make discriminations of colour. This area V4, no homologue of it has been found in any other species below primates. So it looks as though evolution suddenly produced not only the mutation for the extra pigment, but then the whole processing machinery in the brain to interpret colour information. And colour seems to be important, but it's a sort of evolutionary afterthought. Cats can recognise shapes very well. Mice can recognise shapes, although they're very bad at colour. So it was always a kind of evolutionary add-on that colour mechanisms were generated, and that's reflected in the labile nature of the relationship between colour and form. I think it's possible that's why the vast majority of people who have the condition called synesthesia, which I suspect most of you have heard the term, it's a condition in which people experience extra sensory impressions, which, as it were, normal people don't experience. Most of those people experience colour as their extra, so-called concurrent sensory perceptions. The commonest form of synesthesia, for instance, is an impression of colour in the mind's eye, or sometimes apparently projected out into space, when people hear particular words, especially words relating to time, days of the week, months of the year, or whatever. That's really quite common. Probably 2% of the population have that condition. They are really aware. It's not just an association. They're really aware of the colour, either as an image in their mind or projected out into space when they hear a particular word. Other people see colours when they see letters of the alphabet or digits, and sometimes they even see the colour as if printed on the digit. As they read the newspaper, every letter, every number appears to be a distinctive colour as they see it on the page. I, with Megan Stephen, who was one of my graduate students, studied a particularly interesting group of synesthetes who had visual synesthetic impressions, but they were blind. and had been blind, all of them, for at least 20 years. They'd all had vision when they were young, and I'm sure that's significant. But they became blind, and they retained, and even quite clearly in some cases, developed additional visual synesthesias after they became blind. For instance, several of them saw colours when they read Braille characters. Well, clearly that must be related to learning to read Braille, which they didn't do until they started to become blind. So that gives a fix on when the association was developed in their brain, which leads to these extra sensations. This is the impression that, as described to us, that one of our star synesthetes, blind synesthetes, described when he listened to Days of the Week. He said that he saw coloured blobs out in front of him, which represented the present week and the next week. Just 2 weeks were represented. So here was the present week, this was the next week. These blobs appeared to be out. So if you said to him, on Wednesday of next week, he would see this grey patch appear in front of him. The patch, by the way, in his case, didn't move with his eyes. So when he moved his eyes from side to side, the patch stayed where it was. So it obeyed the rules that we apply to the external world. Because when you move your eyes, you don't see the world jumping around. This is a kind of classical problem in visual neuroscience. How is that done? And the conventional view is that some sort of complex and elaborate computation is performed between one view and the next view, and they're kind of pieced together to reconstruct the true positions of objects in space, regardless of eye movements. That, as I'll show in just a few minutes' time, it cannot be the case. Okay, so people with synesthesia, usually 96, 98% maybe of them, have coloured experiences as their extra add-on. And I think that might reflect the late evolutionary origin of colour processing. You can see, at least in some synesthetic subjects, you can see the activation of V4, their colour area, as they are listening to the things which produce the colour impressions, or looking at the things, the non-coloured things that produce the coloured expression. Here, for instance, is our blind synesthete. Listening, well, first of all, here, imagining colours, he can do that, it activates particular region of his brain, but here, listening to time words, days of the week. So he's just listening in the scanner to Monday, Tuesday, Wednesday, or whatever, and comparing that with listening to other words that don't generate colour. So he's looking at the difference. And here's activation of this region, V4, and an associated area, which is thought to be responsible for processing colour in normal people. There's some activation also, by the way, in V1. And this was another blind person who was not synesthetic, listening to exactly the same words, and there were no activations in his visual areas. So the synesthetic impressions seem to be associated with actual activation of colour areas of the brain, at least in some synesthetes. Let me show you a demonstration, and this will work because it's not a movie. Demonstration of the way in which colour is very capable of attaching itself and binding to anything which is even remotely appropriate to the distribution of colours. And this is a technique that some artists, for instance John Piper, Chagall, have deployed in their paintings, taking advantage of the looseness of the relationship between colour and form that can be tolerated in human vision. I want you to look at this image. If you look at the blue dot in the middle, please, for all of these demonstrations. This is a black and white image, a photograph of a castle. Okay. Now, can you just look at that, which is a vaguely castle-shaped picture made of colours. Look at the blue dot and don't deviate your eyes from it. What you're doing is stimulating different groups of wavelength-sensitive photoreceptors in different parts of your retina because of the different colours. So you're producing an afterimage, a coloured afterimage, which is a continuing pattern of activity in your retina resulting from having partly saturated some of your photoreceptors, okay? If I replace it with this, you can see that afterimage, yes? You should be seeing a very vague, let's just go back to this, and then again, keep watching the, how very frustrating. Yeah, you see the vague patch. Now, if you do the same thing again, you can project that. You've seen that afterwards. You know what form it takes. Vague yellow patch, vaguely blue surroundings. You can project it, carry on looking at the dot, onto an image like this, just randomly selected black and white photograph, and it has very little impact on your interpretation of the image. I'm sure you'll agree. Look at the dot, look at the image, slight differences in brightness perhaps, but now do it again. This is the crucial test. You just look for 10, 15 seconds at the blue dot, keep looking at the dot. What do you see? What did you see? You saw an apparently coloured photograph of the castle, yes? Green grass. And as soon as you move your eye, by the way, it becomes black and white again, because what you're doing is moving the afterimage away from the edges and the forms which grab the colour and hold the colour. As long as that colour, which is being generated only in your eye and very diffusely and poor in quality, has something that vaguely matches its form, the colour will adhere and bind to it very, very readily. So something special about colour. What I hope I'm doing is convincing you that the apparent unity and consistency of your visual experiences, is kind of frayed at the edges. It's not tightly correlated with what the early parts of your visual processing are doing, which implies, of course, that what you see is more than simply what's hitting your eyes, and even more than what's being processed by all this mass of visual computational machinery in the back of your head. This simplified picture shows a few of the identified visual areas in the brain of a monkey. This is a rhesus monkey with a primary visual area at the back and all these different regions, including MT, the motion area, V4, the colour area. The general impression is that there are two streams to this, one of which goes down into the temporal lobe, concerned with recognising faces and shapes and objects, colours and so on. The other more concerned with motion, position in space, which goes up into the parietal lobe. And there's quite a lot of evidence that at least some of what's going on up here, also activated by the eyes, is not so directly entering our conscious experience. It's important for deciding how we pick up objects and so on, but we don't, as it were, see it. So, for instance, a bit of evidence for that, injury down here in people in the inferotemporal cortex can produce a condition called visual agnosia, in which objects can't be recognized. In the most extreme form, objects can't be identified at all. They can sort of be seen, colors can be judged, forms can be judged, but people just don't know what the things are. On the other hand, if you ask a person with that condition to pick up an object and then push it through an aperture shaped to the object, they can do so quite accurately, even though they can't recognize that object compared with other objects that won't go through the aperture. So that their brain is able to make discriminations that they, as it were, the observer, can't make. There are also other areas in which processing of information is happening below or beyond the level of subjective experience. This is work done in the functional imaging laboratory, the Wellcome labs in Queen Square, showing that the identification of facial expression, its meaning, its emotional meaning, can sometimes be done subconsciously. In this particular experiment, an image was shown of a face, a threatening face or a fearful face, followed rapidly afterward, that was shown for a short period of time, followed by a neutral face, with the timing such that the neutral face, as it were, blanked out the impression of the frightening face. So when the people who'd seen this rapid series of images were asked what they'd seen, they reported only this, and they denied completely having seen a threatening or fearful face. On the other hand, when the group who did this work, this was Ray Dolan, who was here for the first of the lectures, looked at activity in the brain generated by that particular sequence rather than one which consisted of two neutral faces, they found that the presence of the threatening face caused activation in one part of the brain, deep in the brain, the so-called amygdala on the right side, which is known to be involved in regulating emotional responses. So the brain was detecting that pattern, even though the person was apparently unaware of it. So a good deal of our knowledge and interpretation of the world is happening, as it were, below the threshold of subjectivity. In addition, it's quite clear that what we apparently see is detached in other ways from the reality of what's happening on our retina. This is a nice example, beautiful computational composition by Ted Adelson. What you're seeing is an array of tiles, of checkers here, with an object standing on it which is casting a shadow. The presence of the shadow influences your interpretation of the apparent brightnesses of these patches. So if you compare A and B, it's very clear that B is much lighter than A. A is black and B is white. It's within a shadow, but it appears to be white. Well, in reality, and I think I should be able to convince you of this, if you put a bar in which is a uniform grey bar, which just links the two, you can see that the brightness of this and this are identical. This looks much darker when you don't have the bar to join the two of them together because of inferences that are being drawn by the interpretive machinery in your brain about the relative brightness of the pattern, taking account of the fact that you know that there's a shadow there. Another example that is a very nice illustration of the fact that your interpretation of even of complex things must be dependent on complex binding processes which are influenced by your expectations and your past experience. This is a paper that was published by a colleague of mine, Peter Thompson, who is at York now. It was published in, is it 1980? Yes, 1980, which you'll remember was the height of the Thatcher assault on higher education, when the universities in general were terrorised by the problems of cuts and rearrangements in the universities. He published very bravely a paper called Margaret Thatcher, A New Illusion, and this is one of the illustrations from it. Two pictures of Margaret Thatcher, both apparently showing a cheerful Margaret Thatcher. The difference only becomes apparent when you view them the other way up. Okay, this is the real Thatcher, and in this one, in this Thatcher, the mouth has been inverted and the eyes have been inverted. Okay? But again, if you turn it back the right way up, the way that you saw it originally, it's extremely difficult to see this, which looks like a smiling face, as being this one as being anything but a normal face with a smile on it. And one of the obvious interpretations of this is that the brain machinery responsible for analysing smiles, the shapes of smiles, must be distinctive and segregated from the machinery for analysing the presence of a face. You see the face, it's an upside down face, you see the smile and you recognise it as being a smile, your brain somehow binds those two interpretations together and you see a smiling upside down face. I think very strong evidence of the binding process and the way in which it can be tricked by errors in the nature of the things which are being bound. And just to show there's no political bias in this talk, this is another very well-known politician from a different party, Tony Blair. I saw this headline in The Independent a few years ago when this very famous fossilised footprint was found. In fact, it was a group of individuals, a child, presumably a child following its mother or its parents. It caused a lot of interest amongst paleoanthropologists. So here was this photograph of the oldest footprint, and I thought, well, it doesn't look like a footprint to me. It looks like a sort of set of elevated features in the ground, until I realised that they'd printed it the wrong way up. At least the wrong way up for our interpretive mechanisms. Here we are. Now that looks much more like a footprint to me. Do you agree? The toes here are producing nice holes in the ground, which you don't see if it's viewed the original way up. These look like little sort of lumps in the ground. Again, the usual interpretation of that is that we're accustomed to viewing objects, making assumptions about the direction of lighting. And if you see an image in which the lighting is, as it were, coming the wrong direction, you'll misinterpret the forms of the convexities and concavities in the image. Another well-known example is this. This was an image from the first, I think it was Saturn, wasn't it? It was one of the very early planetary explorers. I'm pretty sure it was Saturn. And this image was produced long-range view from this exploratory probe, and caused a great sensation because people talked about faces on the surface. This thing is about 500 miles across. But the particular arrangement of light and dark and the lighting arrangements on the surface of those hills, mountains on the surface of the planet, it's almost impossible not to interpret it as a face. It's as if we're geared up for seeing faces. We're very sensitive to the presence of faces, which makes a lot of sense biologically. So here, for instance, the face of Jesus found on a frying pan, and here is the offending frying pan, and the inside story in the Daily Sport about that says that the couple, it's nice, the gravel worker who found it said he was discussing with his wife Mary Lou and son Juan Jr. about whether to sell the pan. We might well sell it, we're really not sure what to do about it at the moment. A bit shocked about it. 2 months ago, a woman from Florida who discovered an image of the Virgin Mary in her toasted cheese sandwich sold it on eBay for £14,000. What do you think of this image? Is there anything odd about it? Photograph of a...
Very bright, something that's very extraordinarily bright.
Yeah, not apart from the brightness, is there anything sort of odd about it? And what about this one? No. Does it look real?
No.
Okay, so what do you think it is? It's a model, isn't it? It's obviously a model. And this too, it's a little model scene, doll's house or something. Yeah? Well, it isn't. These are both real photographs. And all that's been done to them is that they've been computationally changed so as to simulate a narrow depth of focus. So the near things have been blurred deliberately in the image, and the far things have been very blurred, much more than you could ever achieve with a normal photograph of a distant scene. You can't get narrow depth of field in a distant scene. It's just impossible. What's remarkable is your brain knows that. So when it sees a scene with narrow depth of field, the interpretation is, this is a model. It must be close to me, because there's no other way that I could have got a photograph of it like this. Well, that is really spooky, because this is an interpretation, a perceptual interpretation, which is entirely based on your knowledge of photography. Showing how much our interpretation of the world is dependent on our experience. I'll just show you very briefly one of a whole class of phenomena, and this was discovered by a colleague who may well be joining us, we hope, in the Centre for the Study of the Senses. If you watch the screen, imagine you're driving a car, and you're looking at the scene ahead. And the road is wet. I know it doesn't appear wet at the moment, but say it's wet, and it throws mud onto your screen. Now, watch the road very carefully, as you would when you're driving, and the mud appears. Did anything happen when the mud splashed onto the screen? Did you see anything strange about the road?
Line going.
How many people saw something funny about that? Some of you have seen it before. This is by Kevin O'Regan, who's in Paris at the moment. We very much hope he's going to come here. Let me just show you what exactly happened. That happened when the splash of mud came onto the screen. So if I can go back, I'll convince you again. There it is without the mud, here with the mud, and the line joins. But you don't see it. You're distracted, even though the event is happening at the same time, which is remarkable. It's not as though something happens and then you blank out something that happens in the past or that happens in the future as a result of the flash occurring. Even exactly the same time, you often don't see it. This implies that your ability to take information from the visual field is really quite limited, and is drawn, that mechanism of extracting information is drawn by the salience of the unexpected nature of what's happening. So let's now very finally consider the problem a little bit more deeply of what information the brain really has to deal with in order to produce what appears to be the image of vision, the detailed, perfect, veridical image of vision. I mentioned that the eye is constantly in motion, and that our eyes have this small patch of really heightened sensitivity and awareness and colour that's zipping around different places. Well, the eye jumps to a new position approximately 3 times every second, even if you don't want it to. These eye movements are happening all the time. You can make them very deliberately if you want. I can look from there to there, deciding to do so. But most of the time I'm not deciding what to do. Some sort of internal algorithm is doing that for me. But my eye is jumping around about 3 times a second. All right, well then, what is your brain experiencing as a result of that? These are some movies that I made with Peter Neary, who is visiting to give a talk here, by the way, next week, trying to simulate what information actually arrives at your brain as you're viewing a normal scene. So what we're looking at first, will the movie work? Yes, good. What you're viewing is the Promenade des Anglais, filmed with a video camera, which is just being carried as if simulating the movements of the head of the person looking around the scene as they're walking along. All right? So this is what your head, as it were, might see if you're walking along this particular scene. However, the eye is also moving. So what your eye sees for any particular view will be only a small patch of this constantly changing scene. Yes, time to stop my lecture, I know. And that patch will be jumping around within the field of view. So, what your retina is, as it were, passing to your brain is something more like this. This is a scene viewed from your head, but with the patch, with your eye movements moving around within the overall field of view. But each point on your retina is connected, as Descartes saw, to a particular position in your brain. So the nerve cells in your brain, which are dependent on what's coming in from the retina to understand the world, are those that are connected to the very central area will be seeing whatever the central area is looking at, and that will be changing 3 times a second, jumping around the scene. So if you can sort of imagine what the visual cortex is seeing, what the foveal representation of the visual cortex is seeing, it's something like this, but it's not because it won't, and I really do have to show you that one. It's my last image. And here it is here. Yeah. And I hope this will work. Yeah. The jitter, by the way, is deliberate because the eye is also vibrating constantly about 30-40 cycles a second. So when you're walking down the scene in the Promenade des Anglais, your eyes moving from position to position, your head's moving around, your gaze is changing. The bit of your visual cortex which is receiving from your fovea will be receiving that information, these snapshots of odd little patches of stuff in the visual scene. And your vision depends on that. It is that information with which you see the richness of the scene around you. So I hope I've convinced you that vision is a remarkable process, and it works after all. We do get around in the world, we do recognize faces, we do make our way correctly to different parts of the room and so on, but an enormous fraction of our impressions are created by our expectations and our past experience rather than what's hitting our retina. Moreover, the evidence from change blindness, that phenomenon of failing to detect things when you're distracted by other things, a whole area of similar experiments suggests that our retention of past information every time we move our eyes is absolutely minimal. That as we move our eyes from one position to another, virtually everything from the past is lost, completely lost. We can't remember it. So this impression of living in a continuously flowing, perfect video of the world is deeply illusory. We have no real knowledge of the past. Well, obviously we have something, because if, for instance, this room were suddenly to change to a completely different room, a cut in a movie, for instance, as you watch a movie, it changes from one scene to another, then you know the scene has changed. So obviously you must remember something about the past in order to know that the new scene is different. But what you remember is absolutely minimal in content and is not of a pictorial form. It's almost certainly of some kind of semantic or descriptive form, more like language, more like room, person, light, colour, rather than a detailed description of the intricacies of what you apparently see. So I hope I've convinced you that although vision is indeed our primary sense, it's a quite remarkable sense. It is not faithfully following the information we receive from our eyes. It is very largely constructed by past experience and expectation. It's easily fooled. And it generates for the fortunate viewer, as it were, the internal viewer of the world, this wonderful, though largely illusory impression. That then leads to really the crux of the problem which philosophers have to tackle, which is, if, as Descartes said, our eyes can provide sufficient information for us to get around in the world reliably, and they do, why do we go to the trouble and the bother of trying to simulate this subjective impression of the world in a way that our eyes are incapable of describing in detail? Why bother with that? All these illusory things which are being created can't be of value in controlling the action. They're not reliable. They're not related to the world. They're pleasant to see. So why do we go to the bother of generating these internal subjective conscious impressions of the world around us and all our sensory experiences, when our automatic and robotic systems have only the real thing to work with? Thank you.